Recall then that free fatty acids are not going to exist alone for particularly long periods of time within a cellular system. They act like detergents, they're not particularly good for the cell. So in fact, the storage form of fats and as well as the dietary form of fats are triacylglycerols. So we recognize that those are the esterified form in which we're going to see fatty acids being stored. That is to say that the long fatty acyl side chains are linked via an ester bond to a glycerol backbone. And this is the common form in which our body, for example, within our adipocytes, that's how we're going to store fats. It's also the way in which you're going to take in fats in your diet. And as we mentioned, fats are packed with power. And some of you are already flexing your sexy OChem minds, are like, I'm looking at these long hydrocarbon side chains and saying to myself, man, those are wicked packed with power. Think about how much energy could be gleaned by ripping and stripping the high energy electrons off of those carbon-carbon-carbon hydrogen bonds. Said another way, think about the power or energy that would be released when the hydrocarbon side chains of a fat are oxidized. Now, Brad will tell us that that is a lot of energy that's released in the fossil fuel industry. That's why we like to tap into resources that provide hydrocarbons. But it's also why our body can glean a lot of energy from these very highly reduced bonds. And what your body does with them is to rip and strip high energy electrons from them, take those electrons to the ETC on those electron carrying molecules, use those electrons to then form a proton motive force. That proton motive force is a pent-up battery-like power that can be tapped into to make our favorite ATP molecule. So we recognize the packed power in a triacylglycerol. However, I want to draw our attention to something else about a triacylglycerol. In the form that we have currently drawn right here, it is a sterically non-committed molecule, right? No, reminds us of people we know, sterically uncommitted, all the best people, right? And so we recognize that in its current form, perhaps we can't really distinguish between the fatty acyl side chains, and we're not necessarily showing a lot of chirality to that number 2 labeled carbon. So you might even ask yourself, how is it that Rachel's labeling that carbon number 2 when in fact you're not able to distinguish? However, if we do look at this with maybe just a little bit of a second view, recognizing that perhaps the hydrocarbon side chain of labeled R1 here is different from that labeled R3, we can start to get a view of something that does have a central chiral carbon. If we were to just take that molecule then and just sort of flip it on its side so that we could begin to look at stereochemistry, we'll draw carbon 2 here as sort of the central point and put that label, this is carbon 2. Let's then draw the bonds that are coming out of the page at you. So kind of fill that in. And then, of course, the bonds that are going into the page and away from you. And recognize then that this is the hydrogen atom bound to that carbon 2. And then, of course, it has its ester linkage. And we'll call this the R2 group, bound to, of course, carbon 1 here. And drawing the ester linkage from this. And recognizing that this R1, if that structure is different at that R1, then this is what contributes to the chirality of that central carbon. And now we can finally add on, make sure we get our full formula there. We can now add on the third carbon, carbon 3, and show its ester linkage as well, and R3. And recognize now the stereochemistry of what was a stereochemically non-committed structure over here, we've now drawn in the stereochemistry, enabling us to see and recognize the ability for carbon number 2 to be a central chiral carbon if the R1 and R3 groups differ from the R2. Then, of course, that central carbon is in fact bound to 4 different constituent groups. With our societal aversion to fats and our desire to try to have something that tastes like fat but isn't fat and that won't contribute to caloric intake for our bodies, there was a craze in the 80s and 90s to develop a fatty tasting potato chip that didn't have any absorbable fat. And onto the scene came Olestra. And this is a picture of Olestra. It's fascinating to look at the structural differences between Olestra and a triacylglycerol. Notice that in Olestra, instead of having a glycerol backbone, it actually has a disaccharide backbone. This disaccharide you might recognize as sucrose. Sucrose is then esterified to count them 8 fatty acyl side chains. So 1, 2, 3, 4, 5, 6, 7, 8. So this is a lot of fat and a lot of slippery, fatty tasting food. But guess what? We cannot break down this epically large molecule. So Olestra slips right through our GIT, and along with it goes all of our fat-soluble vitamins, D, A, and K, for example. So Olestra was brought into question because, of course, it was contributing to malnutrition, and we were seeing some sort of dubious side effects of Olestra, such as anal leakage and very slippery, oily diarrhea. So Olestra fell out of favor, and even Lay's light potato chips were taken off of the market in 2013. There's a lot of very angry customers that you can find online if you're up for a good laugh. So this is Olestra. So when we think about triacylglycerols, perhaps they're the better part of valor. If you really want a fatty snack, go ahead and take in a triacylglycerol, the type of fat that can contribute to energy in us. So we recognize that structurally, these are very, very highly hydrophobic molecules. Remember, all those fatty acyl side chains make them very, very water-hating. That makes them very, very anhydrous. They can be stored in a super lightweight form. And remember talking about how that benefits migrating birds. They've got this super high energy molecule that is very, very light in weight. If I were a hummingbird, I would certainly be very, very glad of my fats. And actually, I'll admit it, I'm very glad of my fat stores every time I do an epically long mountain run of, 3-4 hours or even less. I love my fat stores. So within naturally occurring stored samples, we find as many as 20 to 30 different types of triacylglycerols. And I'm actually going to show you a picture that helps us come full circle today. Remember, we talked about avocados and avocados being packed full of good fat. Well, the types of triacylglycerols that you'll commonly see in avocado include things like linolenic acid. So remember our omega-3. We also would see linoleic acid, our omega-6, and we would also see oleic acid. So that one has a monounsaturated. It is monounsaturated. So this is a very good fat and one that you most certainly want to take in your diet. It's also a good example of how we may see fats stored in our body, so the stored fats as well. So triacylglycerols are the main lipid present in our diets. They are also the main lipid present in our adipocytes, which are fat storing cells. So actually, let's jump down here and write down that adipocytes are fat storage cells. And in a moment, we're going to look at a picture of an adipocyte. They're kind of interesting looking cells, capable of amazing storage. So we know then that lipids are digested in the small intestine, and so it goes to follow that just as we know that detergents emulsify fat, dirt, and oil and grease, right, and trap them into micelles, that's exactly what happens in our gut as well. We have a very special kind of detergent in our gut. It's called the bile salts. Bile salts are detergents. They have a polar head group, hydrophobic side chain, and they are readily emulsifying the fats that we take in our diet. In order to emulsify these fats, we digest them first with pancreatic lipase, and we'll be talking more about the details of this soon, but it actually cleaves at carbon number 1, creating a monoacylglycerol, carbon 1 and 3, and creating a monoacylglycerol and 2 free fatty acids that can then be taken up, can be emulsified by the bile salts, taken up into our intestinal cell lining, and then re-esterified to make triacylglycerols. Lipids then have to be transported through the body, not as free hydrophobic molecules, but as complexes with proteins. These are called lipoproteins. So to just jot that down, lipids are transported through the body as lipid protein complexes called lipoproteins. Now here is a sweet picture of an adipocyte, and of course adipocytes potentially also maliciously malign, because we know that the hypodermal, like subsurface skin surface layer of adipocytes can certainly grow, and that can lead to obesity, but we also recognize that there's a certain amount of fat around your viscera, and if you don't have enough fat to cushion that viscera, then that can actually be extremely deleterious. So as I talked about a couple of my athletes having under 3% body fat, that we start to worry about them having that tissuing capacity, that cushioning capacity. We also start to worry, as I mentioned earlier, about thermal regulation. So adipocytes playing a big role in that as well. Kind of a cool figure, huh?